I'm Natalie Sims. I'm Deputy Director of St Vincent's Institute and Head of the Bone Cell Biology and Disease Unit, and I'm also a Professorial Fellow at the University of Melbourne and at Australian Catholic University. I didn't set out to be a scientist. I did a science degree, and at the end of the degree chose to do an honours year, which is a one-year research project, and that was on the effect of dietary calcium on the skeleton. And during that year, which was with Howard Morris at the Institute of Medical and Veterinary Sciences in South Australia, I found out what research was like. I realised that research was a living, breathing thing, and it was something I could do and something I enjoyed. And so then I decided to do a PhD. So that was a real turning point for my whole life, because I hadn't even considered doing research as a career before that. The other major event that was a turning point was when I did postdoctoral studies in the USA at Yale University with Roland Barron. And there I was in an environment where there were many different projects I could work on at once. It was the start of the use of genetically altered mice in studying bone biology, and he threw lots of projects at me. So I got to work on many different things, and really it was a huge growth in my career, a massive period of learning to work out how to look at the structure of the bones of these mice that had their genes knocked out for a range of different agents, and I could see that there was lots to be done. The easiest way to sum up my work is bone cell biology. So I look at the cells within bone and how they control bone structure and bone strength. We use a number of different techniques. Typically, my work has used a lot of genetically altered mouse models where we knock out a gene for a protein to find out what it does to the structure of the skeleton, and then we look at that structure either down the microscope or by other imaging techniques and see how that's changed to then find out what that protein does to bone structure. More recently, we've also started looking at the internal structure, the material composition of human bone, and seeing how that changes with ageing. My work's largely funded by the NHMRC with some ARC funding, but over the years, because NHMRC funding doesn't really cover the full cost of research anymore, it's very dependent on philanthropic funding as well. So here at St Vincent's Institute, we have a foundation that people donate to, and that also supports my research along with many others in our institute. There's probably two things that drive me in my work. One of them is that bone is really fascinating. I don't get tired of learning new things about how the skeleton works, what it looks like. It makes me very happy to just look down the microscope and see the structure and know that there's a lot we don't know about how the skeleton works. So part of it is this passion for discovery and wanting to learn more. The other part that's really important is knowing that if we learn more about how the skeleton functions, we should be able to find new ways to prevent fractures in people who are likely to have them. So it's finding out what makes bones strong so that we can manipulate that and help people who are ageing to be able to keep their bones strong throughout the rest of their life. Yeah, I play a lot of music outside of work, so I sing and I play violin and piano. I love music, it gives me so much joy. And I get to enjoy that with friends and my community and sometimes with my family as well.